Friends, if you remember the earlier two lectures that we had on photosynthesis, the first one was devoted at great length to the history of the development of the concept and the formulation of the equation, the right equation for photosynthesis. The second lecture was devoted entirely to the fate of light. Of course, also having a discussion on the nature of light, where we had solved all different types of physical constants. We had talked about the different photosynthetic pigments, their properties, and highlighting chlorophyll A as the principal universal pigment, and the properties which were coordinating the whole important phenomenon of photosynthesis as far as the near overlapping of the action and the absorption spectra was concerned. Then we had also reached a conclusion that the process of photosynthesis in fact is made up of two main parts. The light reaction, as you can see on the screen, the main function of light would be to activate chlorophyll, which we technically call as photoexcitation of chlorophyll. As a result, water is going to be split or broken down, which we would technically call as the photolysis of water, and as a result a lot of energy would be released through intricate manner and through the electron transport chain and photophosphorylation. This we would see today, and as a result oxygen would be released as a byproduct. The protons which would be released in the process of photolysis of water would now be required to reduce the coenzyme 2, that is NADP plus, to give rise to NADPH, and the extra amount of energy would go to the so-called ATP synthetase in the phosphorylating particles embedded on the membrane of the grana to produce ATP. So that means the two products of light reaction would now be required for the running of the Benson-Calvin cycle, which would constitute the dark reaction. Whereas the light phase or light reaction is operative in the grana, the stroma is the site for the dark reaction or the so-called Blackman reactions, where a carbon dioxide would be taken in, and through an intricate process of metabolic pathway, ultimately sugars are going to be released. And once these sugars are going to be formed, they have different fates. They could be conveniently stored if they are not required, or they would be instantly metabolized. So this was the basic background of the process. And we had also in our second lecture focused on what are the different forms of light which can be useful to the process of photosynthesis, because not every form of excitation is or the release of energy is actually useful for the process of photosynthesis. One thing is very clear, that the chlorophyll molecules have been photoexcited, and once they have reached an excited state, they have to come back to the ground state. And if they do not react chemically before that, then they will lose their extra energy, and that would be a wastage. So there are basically four ways by which the extra energy can be dissipated or lost. The question is, which one of them is useful to drive the process of photosynthesis, in this case the light reaction. The energy can be released in the form of heat, light, or even inductive resonance, which is a kind of a radiation-less transfer. We learned that these three forms may not be that useful for driving the process of light reaction. So it is basically the photochemical reactions which are the useful ones, which would mean somehow when the chlorophyll A molecule is photooxidized, there has to be some acceptor molecule which would accept these electrons, and it would get reduced. So that means out of the four alternatives that we had mentioned, maybe it was fluorescence or phosphorescence or heat loss at different levels, it is basically the photochemical reaction which is the quickest of all the four, and as we would see subsequently, it is the most useful reaction for the initiation of photosynthesis. Now, it would mean that the light reaction would include three important parts. The first part, which we will study in details, is the photoexcitation. How is it brought about, how the photons are absorbed, and how ultimately it results in the splitting of water and release of electrons. Because if the release of electrons is there from the reaction center, then only the further progress can be made in the process. And this is by number 2 part, and that is the electron transport. So that means there has to be a well organized electron transport system which would be present on the membrane. What are the different components? How are they arranged vectorially on a membrane? Which face are they towards? That is, whether they are pointing towards the stroma or they are towards the lumen side. This has to be seen. And whenever the protons are going to be formed, where exactly is the concentration gradient of the protons, which would drive a proton motive force, which we would see in the part 3, and that is the photophosphorylation. The term photophosphorylation reminds us of oxidative phosphorylation, which we study in details in respiration. Of course, needless to say, there are many similarities between the two processes. However, there are a few fundamental differences too. So, when we talk of photoexcitation, we should remind ourselves that when a chlorophyll molecule absorbs energy from sun in the form of photons or light quanta, earlier the chlorophyll was in a ground state, and now it is suddenly excited, and this is such a quick process which would say run to about one by a millionth of a second, and it would mean that these excited electrons have to fall back on the ground unless they meet somebody to run away with. That means to get transferred, and if they don't, the energy, the extra energy which will be released in the process, as I said before, would be in the form of heat or light. So that means they have to be transported and to be transferred to a proper electron acceptor in order to have a useful role in the process of photosynthesis. And our recent ideas tell us that this type of an energy transfer is mediated through specialized reaction centers, specialized antennae which are in a group. This we would also see now, and the light which is first falling on the antenna complex of the pigment molecules ultimately is going to transfer their own respective energies through resonance transfer, which we call as the radiationless transfer, and finally reaching the reaction center. And it is from the reaction center that so much of energization of this reaction center has taken place that it is able to eject the electrons, and through a well-defined electron transport chain, it brings about the future reactions. If we remind ourselves of the position of chlorophyll, we would see that pigment molecules are located on the thylakoid membranes, and they would be arranged in the form of an antenna. An antenna would be which could somehow concentrate a huge gambit of these photons and bring them to a focal center, which in this case would be the reaction center. and once the light strikes the antenna complex, it has to be channelized towards the reaction center, which is the focal center. And when enough energy is present there in the reaction center, then the electrons are going to be finally ejected from there. One thing we know for sure and for a long time, that one photon of light is considered to be able to release one electron. So this kind of equivalence is going to be the basis of our subsequent calculations. But then let us now start the whole idea by asking ourselves a very important question. The whole equation of photosynthesis revolves around light, oxygen being released, and water being photolysed. So the question is, how many quanta of light are really necessary for evolving one molecule of oxygen? In the balanced equation, we see that it is 6 molecules of oxygen which are being released. So what do we put as the answer to the number? Whether it is 6 photons, or is there a multiple of 4 or 8 or 10? This has to be understood. And this can be understood only on the basis of certain constants and certain terms. The first one is quantum efficiency. Now, what would this quantum efficiency mean? It would mean what is the amount of the light quanta required for the process, and the quantum yield would be how much amount of oxygen is released per light quanta. So that means these are the two constants which we have to first understand. Quantum yield, or as we abbreviate it by the letter phi, it's measured as the amount of oxygen evolved or the amount of carbon dioxide taken in. Now, the amount of carbon dioxide taken in is a little shaky business, so we refer only to the oxygen, and we say it's the number of oxygen molecules released per photon. Number of oxygen molecules released per photon. So that means it is this quantum yield which expresses the efficiency of light whenever a photon is absorbed by a molecule. Whereas quantum requirement is inversely proportional. It's opposite to the quantum number. So it is 1 by phi. And it would mean what is the number of photons or light quanta which are required to release one molecule or to reduce a CO2 molecule. So that means both these constants are frequently used, but we generally talk in terms of the quantum yield. And the quantum yield would be now what is the number of oxygen molecules which are released per light quanta. If you remind ourselves of Einstein's law of photochemical equivalence, it has been said many years before that whenever there is a photoelectric effect, we find that one hit of a photon on any molecule is going to lead to the expulsion or the release of only one electron at a time from the molecule. So this type of an equivalence is there with us, that is 1:1 photon. One photon equals 1 of the electrons. In the present equation that we had on photosynthesis, we find that 4 electrons are needed to be extracted from a water molecule for the evolution of one oxygen molecule. Why is it so? Because we find that 4 hydrogen atoms are required to reduce one molecule of carbon dioxide. Indirectly, 2 molecules of water. This we can confirm on the basis of the equation which is on display right now, which means that when 4 molecules of water are going to break down into 4 protons and 4 hydroxyl ions, the 4 hydroxyl ions would give oxygen, and in the process they would also release 4 electrons. So that means this would be a term which would be called as 4 H ions, 4 protons required per oxygen. The initial work of Warburg had given us the idea of the quantum requirement of 4. Now we have to be very careful, because Warburg was the person who initiated all these studies, and he was a pioneer in the field, and he calculated that 4 of the protons are necessary. This idea of quantum requirement of 4 would suggest to us that photosynthesis is a very, very efficient process, extraordinary efficiency. Then we ask ourselves a very pertinent question: are the biochemical processes extraordinarily efficient? Is there no process which is wasting energy in the intricacies of the metabolism? So subsequent work was done by Emerson and Arnold and other workers, and they found out that the quantum requirement was actually not 4, but it was 8 to 10. Now this was a remarkable discovery, because it meant that the photochemical process that we are discussing right now is not in fact a single step reaction. It is in fact a reaction where each of the electrons needs to be excited twice. This would mean that if an electron needs to be excited twice, how can we take the figure of 4 as the quantum requirement for the process? If we go further into the energetics of the process in order to disprove what was the opinion of Otto Warburg, we would find that one molecule of glucose contains about 686 kilocalories, which means conversion into kilojoules would be 468.6. And by that analogy, if we take 1/6 of glucose molecule, which would now contain just 1 oxygen, so this possesses 112 kilocalories. So if this is so, we have seen in our previous lectures that the best and the most effective wavelength in photosynthesis is the red light. And the red light photon contains about 40 kilocalories of energy. So if we are out to calculate the efficiency, it comes, the efficiency of conversion of light energy into chemical energy comes to 112 by 40, and then you multiply by the quantum requirement 4, it is 70%. Now 70% seems to be unbelievably high. A process we sincerely wish photosynthesis was 70% efficient. So that means there would not have been any problem of or any food crisis. The amount of matter which would now be fixed would have been tremendous. So it is not so. Photosynthesis is certainly not as efficient so as to convert the entire light energy into chemical energy. Forget about a biochemical pathway, even no machine can show 100% efficiency, because there is a considerable loss of energy during the process, which we had seen earlier also. A lot of energy is lost as heat and useless forms of light. So that means if we are going to take the modern concepts into consideration, the quantum requirement should be taken, say, between 8 and 10, and now the quantum yield would be, as Warburg said, it was going to be 0.25, but then from the modern point of view, we don't have to divide 1 by 4, we have to divide 1 by 8 or 1 by 10. So that means now the whole idea of the quantum yield would be 0.125. So that means our ideas or the figures have been reduced by almost 50%. So this is the first part as to how many of the light quanta are required to produce a molecule of oxygen. Certainly they are not 4, although theoretically they appear to be 4 with the help of the equation. But then if you are going to do something twice, then you have to spend twice. It's as simple as that. Emerson and his co-workers, they had some classical experiments to perform, and they did it in 1943 on the unicellular green alga, which is again the wonder material for photosynthesis, chlorella. What they observed was that while they were performing these experiments, if they had exposed chlorella to wavelengths which were greater than 680 nanometers, now this comes in the far red region, there was a sudden drop in the quantum yield. You could clearly observe the absorption spectrum of chlorophyll, which suggests that there is one peak at the blue region and the other at the red region. But then when we were going beyond red, that is in the far red region, beyond 680 nanometers, there was a sudden decline. That means the value of phi was really going down, which meant there was some sort of inhibition. So this indicated that the area of the region beyond red was somehow not very conducive. It was less efficient, and this phenomenon was called by Emerson as red drop. So this is a phenomenon which really helped us to discover what we call as the two pigment system of photosynthesis, or the two photosystems. Later experiments by the Emerson's laboratory, they also performed certain experiments which were even more interesting. They found out that if there was a drop, or the so-called red drop, you could even restore the whole photosynthesis, or that means you could abolish this red drop, provided the two beams of light of different wavelengths were given. That means one was red and the other was far red. It could be red or even lower than red, and if these two were given simultaneously, immediately photosynthetic rate picked up and it was restored to normal. Not even normal, under two simultaneous or superimposed light beams, it could even be 2 or 3 times more than the sum total of the rates which were obtained by two beams individually. So this was a sudden enhancement in the rate of photosynthesis. If one sees the figure, which means if the far red is on, there is some photosynthesis, and after switching it off, again a flash of red light would mean some photosynthesis. But if both the lights, which means wavelengths less than 680 and the ones greater than 680, there is a sudden spurt in the rate of photosynthesis, and there's a tremendous enhancement. It was a very good evidence that these two photosystems, that is, why did we call the term photosystem? Because it was being, the electrons were being excited twice, so there must be two different areas or adjoining areas which were doing this business, and they were working in tandem. So this in honor of Robert Emerson, the name was given as, or the term was given as, Emerson's enhancement effect. Now this was more than a conclusive evidence that it's not only one, but there are two photochemical reactions which are in fact involved, and we cannot see them in isolation. They have to be seen together. And these two photosystems are the ones, that is, one is driven by a short wavelength which is less than 680, that means it will be high energy one, and the other which is a long wavelength one. So that means in order to ensure that the process of photosynthesis takes place optimally, both these systems have to be driven simultaneously, or at least in rapid succession. So till now we have made one thing very clear, that there are two photosystems. Another very interesting result came from the work of a much earlier work by investigation by Emerson and Arnold. They said that Okay, if chlorella is working in terms of a photosystem, then there should be a saturation which would be proportional to the number of chlorophyll molecules. So what they did was, they were finding out the quantum yield, and they found out that after giving a flash of a few milliseconds, the saturation was achieved, even though 2500 chlorophyll molecules were present. Now this was a very unexpected result, which meant that if we theoretically expect the saturation when all the chlorophyll molecules are in use, it should go much higher. But then there is a saturation when photosynthesis is used. This is the observed one, which is much lower. What does it mean? It means that perhaps all the chlorophyll molecules are not working. There is a distinct division of labor, and all the chlorophyll molecules are not doing the same thing. Now this was a very important conclusion, and Emerson and Arnold said it definitely means that light is absorbed by a group of pigment molecules acting as a single unit. So that means this is the 2 units or 3 units, whatever they are. We have concluded there were 2, that this was the concept of photosystems. So that means it is not a random functioning of the chlorophyll molecules. They must be arranged in a group performing a specific job. You're all familiar with the concepts of quantasomes in cell biology, and it has been widely felt, it was at least a few years earlier, that quantasomes are the photosynthetic units. We ask ourselves again a question: are they really the photosynthetic units? Let's try to find out the answer. The earlier ideas, as we said, according to Emerson and Arnold, a photosynthetic unit was 2500 chlorophyll molecules acting as a single unit, and they would be releasing 1 oxygen molecule. So that means somehow the equation started emerging, and later ideas were entirely different. They said that 8 to 10 photons are there per molecule. So people logically assumed that each photosynthetic unit must comprise of 250 chlorophyll molecules, because 2500 are taking part and 10 photons are required to release 1 oxygen molecule. The story was pretty good and well knit, until 2 cytologists, Park and Biggins, they found out by a specialized technique of freeze fracture that there were certain granules in the membranes of the grana, and the distribution of these granules was fairly uniform, fairly regular in the grana membrane, and they immediately thought that these are the units that people have been talking about. They must be clusters of 250 chlorophyll molecules plus 50 carotenoids and other accessory pigments of chlorophyll B, and they called it as quantasomes. The story fitted very well, but then the modern ideas negate this whole idea. They say that it is not in the form of a granule, or it is not just chlorophyll molecules. There are so many other ingredients also involved. So it should be called as a light harvesting complex, and that should be the unit of photosynthesis, where chlorophyll A, which I have been telling right from the beginning, is the hero of the whole process, which is the reaction center, because it has to ultimately transfer the electrons to an electron or acceptor in the electron transport chain. And of course, who is going to trap the photons? The antennae. That means all the other participating accessory pigments, including chlorophyll B, the carotenoids, they will work together in unison to trap and then transfer energy to the chlorophyll. So it is something like a funnel. If we see a diagrammatic representation of the so-called reaction center, then the photons would fall on the antenna molecules. There is a transfer of energy, which in this case is resonance transfer, and this all has to be done before the electron can return to the ground state. Once it returns to the ground state, it would again require an expenditure of energy or energization, and ultimately then the energy reaches the reaction center, and it is this reaction center which gets energized and then loses its electrons to the primary electron acceptor, which accepts it, and then the subsequent reaction continues. So under these circumstances, the concept of quantasome now appears to be quite unconvincing, and we find that it is not chlorophyll and chlorophyll alone which we said as the hero. It requires all other actors too in having the whole drama of photosynthesis. So that means the chlorophyll molecules have to cooperate with other ingredients of the reaction center. Let us have a more detailed discussion on the two photosystems that have been discovered on the basis of Emerson's and Arnold's work. The photosystem 1 is they are driven by two systems. The one wavelength less than 680, this is photosystem 2, and wavelengths greater than 680 nanometers, which is the low energy photosystem 1. And each of these photosystems would be having their own antennae, their own reaction centers, their own individual specialized proteinaceous carriers, electron carriers, apart from metal and the non-metal ions. So that means each of the pigment systems or photosystems is unique in itself. This is how we can visualize the organization of a photosystem, where they are embedded in a membrane, and the light harvesting complexes are present which will take up the photons, and through the resonance transfer there is a transfer of energy finally to a special pair of chlorophyll A molecules, which would constitute the reaction center complex. And it is from here that the electrons are going to be ejected, and they are going to be accepted by what we call as the primary electron acceptor. If you look at it carefully, this is the interior of the thylakoid. So that means this is the lumen, and this is the stroma side. So that means we have to also see the vectoral arrangement of all these light harvesting center and the other protein molecules. How are they present on the membrane? So the two photosystems have been biochemically isolated by specialized techniques of density gradient centrifugation, and what we find is that the photosystem 1, or we now abbreviate it as PS1, is constituting the upper fraction because it is lighter, and the chlorophyll molecule which is in the reaction center would be called as trap 1, and there is a specialized chlorophyll A molecule which has the maxima at 700 nanometers. So we nickname it as P700. This is surrounded by nearly 200 light harvesting molecules, which are again greater than working at greater than 680 nanometers. So they are called as chlorophyll A 683. In addition, there are chlorophyll B molecules and the carotenoids, which are going to absorb the light quanta at different wavelengths. In addition, there are many electron carriers, namely cytochrome F, plastocyanin, and two very specialized cytochrome B 563, along with several tyrosine residues, ferredoxin reducing substance FRS, and several membrane bound iron sulfur proteins. So these are the constituents of the lighter fraction, or the PS1. The PS1, as we said, is the upper fraction, and if we compare its chlorophyll A content, we find that chlorophyll A is more, the proportion is more as compared to that in photosystem 2. However, the amount of chlorophyll and carotenoids is more in the PS2, and this PS1 is going to absorb light wavelengths at greater than 680, so naturally it is working at a relatively low energy level. It is because we had the ferredoxin reducing substance, this PS1 is a very strong reductant and a weak oxidant. It is at this site that NADP, that is the coenzyme 2, phosphorylated one, is converted into the reduced form of coenzyme 2, which is NADPH plus the protons. So PS1 is capable of operating independently. You could ask a very relevant question and a logical one. Who gave us the liberty to call this as 1 and the other one as 2? Is it only on the basis of lighter and the heavier one? Perhaps no. It is on the basis of their antiquity, which means now we believe that PS1 originated first. We would like to discuss this subsequently, because what type of photosynthesis is called as the primitive one, which was there, or the so-called anoxygenic. There the pigment system or the photosystem 1 would be working by what we call as cyclic photophosphorylation, and the PS2 or the photosystem 2 originated later, which was capable of releasing oxygen. So photosystem 2, which we believe came later, is the heavier fraction, and its reaction center is again a specialized chlorophyll A molecule, which is working at 680 nanometers. So we will call this as trap 2 or nickname it as P680. And then there are the surrounding chlorophyll molecules of 683. So altogether this functions at less than 680 nanometers. It's a high energy one. And as we said earlier, the chlorophyll B and carotenoids are more. In addition, there is an electron donor Z and a primary electron acceptor Q. The various plastoquinones, and this PS2 is rich in manganese ions and chloride ions. Contrary to PS1, PS2 is a very strong oxidant and a weak reductant. You remember, in PS1, by virtue of being a strong reductant, it could reduce NADP. But here, since the PS1 is a strong oxidant, it oxidizes water. As a result, molecular oxygen is released. So if we were to just graphically talk about the antenna molecules and the light harvesting center, we somehow assume that these antenna-like molecules give us the appearance of some type of a funnel, which we would see subsequently. And these two photosystems are arranged sequentially, and we find that this is how the transport would take place. So that means twice we would have the energization. The PS2 is going to be energized as the heavier one, and water is going to be broken down to release protons and oxygen, and these electrons will pass through an intermediate cytochrome system, which is composed of rice cake proteins also, and then ultimately coming to PS1. The PS1 is also going to lose its electrons, and it's also going to reduce the NADP to NADPH. So this is how a strong oxidant and a strong reductant are functioning in tandem. The antennae which are present are also very closely packed. Why is it so? In other words, why do we need these antennae in the first place? The idea is to concentrate. The idea is to optimize the transfer of excitation so that these photons, once they are trapped, they should not be lost. And then to optimize the transfer of excitation energy, and because ultimately inductive resonance has to take place. So unless these antenna components are closely packed, how could we ensure that there is minimum wastage of energy? Because the main aim or the ultimate aim is that the energy should eventually reach the reaction center, because it is that reaction center which is the site for the photochemical oxidation reduction reactions. So as I said before, it all gives us an idea that it is a energy concentrating machinery of whatever energy is available. So that means the core of the chlorophyll molecules, the reaction center and harvesting molecules must be arranged in a specialized design which gives us a funnel-like situation, because it is only a funnel which can ensure a continuous availability of excitation energy, and it should be highly efficient also. In spite of such a good logistic architecture, 10% of the energy is still lost out of the available one during this inductive resonance, because closely packing or close juxtaposition does not mean that there are no spaces between them for even an electron to pass through. So that means some amount of energy is lost. And this is a graphic representation of how a funnel-like design of the light harvesting center can be conceived, because the 100 molecules of the chlorophyll B which are constituting the antenna would ultimately funnel down to chlorophyll A molecules, which are again the chlorophyll A molecules, and to the chlorophyll A other chlorophyll molecules of different wavelengths, and finally arriving at what we call as the PS2 reaction center. And likewise we have a special pair of chlorophyll A, which is the P700 or the energy trap. So this is the trap 1 and this is trap 2. So the reaction center would, as you can see, right from the shorter wavelengths ultimately to the longer wavelengths, because energy is dissipated always from a more concentrated high energy to a low energy site. So a funnel-like conception of the light harvesting center can explain how the electrons ultimately reach the centers for their subsequent journey to the electron transport system. To understand this, what are the light harvesting centers composed of? Is it only the chlorophylls? No, there are other components as well. So that means the two photosystems actually belong to the category of integral proteins. That means they are embedded in the grana membrane. Once they are embedded and they are integral proteins, they must have a characteristic orientation also within the thylakoid membrane. So that means one of these ends needs to be projected, or it needs to be projecting either into the stroma or towards the lumen. Are all of them pointing towards the same direction, or is there any vectorial heterogeneity in these 4 different complexes that we are talking about. So it is now seen that proteins of the complex 1. Now the complex 1, which is the starting one, is light harvesting 2, which is the photosystem 2, and the complex 2, which is the cytochrome B and F, they are directed towards the lumen. That means the interior of the chloroplast granum. On the other hand, the complex 3, that means it would come later, the photosystem 1, along with the complex 4, from where we have the CF1 and CF0, which are the coupling factors or the ATP synthase, they are directed towards the stroma. Now this is a very interesting aspect of the vectorial arrangement of the complexes. 2 are facing one side and the other 2 the other one. So the vectorial arrangement tells us many things. If we look at this diagram very carefully, you would find that the complex 1 and 2, they are facing towards the lumen, that is the interior of the chloroplast granum, and water is being broken down, protons are being released, oxygen, molecular oxygen is released, and so is the cytochrome B and cytochrome F. It is also directed towards the lumen, and both of them are releasing protons, and they are connected, these 2 photosystems are connected through plastocyanin, and the PS1 along with the ATP synthase complex, they are directed towards the stroma. They are directed towards the stroma. Now this gives us a very important food for thought. As you can see, ultimate products of light reaction which are needed for running the Calvin cycle, and Calvin cycle runs in the stroma, they are produced on a phase which is in the stroma itself. The problem of mobility is not here for ATP or NADPH. The problem is of these protons going out and the components of the PS1 getting into the PS1, that is from PS2 to PS1. For this also we have the other arrangements which we will see. So that means there is a vectorial heterogeneity and there is a lateral homogeneity of the light harvesting centers, in the sense, if we look at a diagrammatic expression of the type of light harvesting centers which are located on the different faces, you would appreciate that this end, that is, there are some ends which are free. Free in the sense they are free towards the stroma and there is nothing to press upon them. So they are called as the unstacked regions or the so-called non-appressed regions. And PS1 and ATPase, as we saw in the earlier diagram, more of, or rather most of these PS1 and the ATP synthase complexes are localized here. Whereas on the stacked regions or the so-called appressed regions where the membranes are facing each other, then we have an abundance of PS2. So PS2 is more on the appressed regions of the chloroplast as compared to the non-appressed regions where ATP synthase and the PS1 exists. As I raised a question as to who is going to carry the intermediates from one photosystem to the other through the membrane, it cannot be an integral protein, because an integral protein, the position or the polarity or the orientation is fixed. So in order to do that job, there are various mobile elements which are also present in the light harvesting centers, and they are the plastoquinones, the plastocyanins, the ferredoxins. So that means their job is to deliver the electrons from the light harvesting center 2 to light harvesting center number 1. and it means that they have the unique capability of diffusion from the grana to the stroma. So certainly by definition they cannot be any integral part, nor can they be permanent part of any of the complexes. That would hinder their mobility. It would hinder their mobility of movement. If we look at how the whole process of excitation takes place, now this is one of the most interesting and the most intriguing aspects of the photochemical reaction, which is called as the exciton transfer. The exciton transfer or the electron transfer. If we look at this figure very carefully, we find that there is a reaction center which is in the center, and these are the different antennae which are giving the idea of an umbrella or a so-called dish antenna, where we have to concentrate and ultimately go to the focal point, which is the reaction center. So that means energy of a photons is going to be transferred through the light quanta, through various accessory pigments and the antenna molecules, ultimately to the reaction center, and each of the reaction center is present in the grana. And this is how the different processes take place. Let us look at it in sequence. The antenna molecules, how are they going to transfer the energy? When light is going to excite an antenna molecule, we would find that its electrons are going to be raised to a higher energy level as compared to the adjacent one. So once they are raised to a higher energy level, they can now pass their energy to the neighboring chlorophyll molecule. And once they have passed this energy to the neighboring molecule, we would find that now the original antenna molecule has its electron back in the ground state, whereas the one which is now shown with an artisac, its electron has now reached the high or the excited levels. Once this has been done, this whole process goes on in succession till the antenna molecule which is adjacent or juxtaposition to the reaction center, it comes in contact with the reaction center. We find that the reaction center molecules have their all these electrons in the ground state. This reaction center receives now energy from the antenna molecule. Now it has to be energized in the sense that the excited reaction center now passes an electron to the electron acceptor. The red ones are the electron acceptors. This is the reaction center, which is blue. You can see the moment the electron is being accepted by the acceptor has turned from plus to minus, and the one which has donated has now become plus. This is the basis, what we call as the charge separation. This is the exciton transfer, which is the basis of the beginning of the electron transfer in photosynthesis. The current idea as to how all these four complexes are temporally and vectorially attached to the membrane and how do they function have to be given a diagrammatic and a functional representation, and that is in the form of a Z scheme. The term Z scheme is not new. It comes from the American word the Z scheme, which means Z or a zigzag scheme. Now this scheme was originally proposed by Robin Hill, but then we are now building up the whole new concept on the basis of this Z scheme, which was proposed by Hill, and the two photosystems have to be fitted. So that means once the electrons are going to be lost from a photosystem, they are having two options. These two options have to be arranged in the form of a Z scheme of photosynthesis. The current work has been pioneered on the modern aspects by Professor Govindji, and the latest scheme we will discuss in details in one of our next lectures as to what are the different options when the PS2 loses its electron, whether it would ever come back or whether it would never come back. So both these stages will constitute what we call as the cyclic and the non-cyclic transfer of electrons, which would have their own implications.